unique, meaning things that only happen during prophase 1 and don't happen any other time during meiosis. That's the thing you should get from this question. Unique to prophase 1 is things that don't happen anywhere else, especially prophase 2, which means they eliminate these possibilities. Now overall, it's not bad that only a few people put them in there, but it's something you want to look at and pay attention to. Only there are some qualifying words that will change what your answers are supposed to be. Now, we all know about probabilities and the calculation of probabilities, right? And what's the probability of getting 4 answers correct? It's less than getting 1 answer correct. That's why the average is pretty low, 25%. Because not everybody picks all the answers. And some people pick weird answers or wrong answers. That decreases the overall beauty of the question. But please take note of some of these things when you're reading. So it's very easy to sort of gloss over a question and say, oh, prophase 1. Now, if I'm doing things correctly and not making any mistakes, the point total of the question should give you an idea of how many things to pick. But you can see, right? You can see the point total before you answer the question. Does it tell you how many points the question's worth? Okay. That's sort of to help you sort of gauge things. The other question that was most commonly missed is this one here. And you can't read it because you have bad eyes and you're too far away. It's about non-disjunction of meiosis. And you can get normal gametes from non-disjunct events, but it'll only happen when non-disjunction occurs during meiosis 2. Now some of you may have misread or interpreted this question. It doesn't say that all of the gametes will be normal, because you can get some gametes that'll have the normal chromosome or the appropriate chromosome number. So most people chose the one where you won't get any normal gametes anytime, whereas a third of you chose the other answer. Any questions about anything meiosis related before we get into some of the calculations? All right, for the first one, where you were dealing with blood types and trying to do just a simple probability, overall the average you get was 73. A lot of people got all points, but there's a scattering of things that bring the average down. People that got the wrong answers, I tried to figure out what you did wrong, but a lot of times I couldn't. I think a lot of times people treated this like the allele frequency calculation problem and doubled the number of individuals rather than choosing it as it is. If that's what you did, I'm sorry that this might have tricked you into doing something wrong. I wasn't trying to be misleading, just trying to give you an easy problem. Here, look at this stuff. Do an easy calculation of probabilities. But take a look at your answers, and hopefully you have some maybe notes where you wrote things down. Bring them to my office. We'll talk about what to do if you got the question wrong. But I don't have a whole lot to say about that quiz. For whatever reason, you maybe can tell me, one of these types of questions was more heavily missed than the other type of question. Does anybody want to guess which one? The or the addition rule questions, or the multiplication rule questions? Which one do you think was missed more often? Multiplication. It was the multiplication. I don't know exactly what people did wrong. Kind of hard to tell sometimes. Some people got close, but it's not the right answer, so I really don't know what you did sometimes. Some people got partial credit because it didn't give you the right number of digits. Punitive with digits. So, sorry about that. But you still got credit, just not full credit. Sorry. For the quiz 4B, it says they were only over 2 points. Wasn't really a point of taking off a point, or just giving me 2 digits instead of 3 digits. But those 3 digits allowed me to know if you actually did the calculation correctly, because some people got 2 or one digit correct, and not all three. But I wonder why these were so bad. Did people just do them using the and rule rather than the then rule? Just let me know what happened for you. Or just not worry about it because you're gonna do better next time. Next time being right now. Because let's do a calculation. I know you're all
Sorry.
calculation. Let's do another practice calculation. I'm sure we're all, for the most part, understanding things. Rather than write on the board, I'm going to write on a piece of paper. Once you think you have the answer, you can come up and look, see if you got the right answer. We'll spend between 5 and 10 minutes with this. Can't spend forever with it, but one thing to keep in mind is this is not the only thing on your test. It's not the only calculation that'll be on exam 2. One thing you want to try to practice is speed and efficiency. You just don't have a whole lot of time to spend on some of these things. You know where you can do shortcuts. These can be much quicker. This one is a little bit easier because everything is heterozygous, which means you should be able to predict what the patterns or ratios are without having to do a bunch of squares. It'll tell you in the exam there will be a few homozygous states. You can't just follow the habit that everything is always heterozygous and heterozygous. What we call classic Mendelian examples. Who's close? Number equation. Luckily, you also gotten this far. So if you get from 1/16, we have to combine the long and the white together. Long's recessive, and heterozygous, you should get 1/4 long, and also 1/4 white. Because homozygous recessive and incomplete should be also 1/4. So 1/4 times 1/4, 1/16. For the 3/8, we look at lavender is 1/2 with the incomplete dominance because it's the intermediate state, it's heterozygous. So 2 of those out of 4, 1/2. And then green is the dominant case. So for the complete dominance example, we would get 3 green, 1 blue, so 3/4 green. Combine those together and get 3 out of 8. And once you have it, you can solve for everything. You just have to make sure you do the math correctly. Depending on how you want to write the answer. Rating powers are 10 as percentage is kind of weird. Just to put the answer out here for everybody to see. 5 digits here rather than the typical 3. That should be your answer. Does anybody have any questions about how we populate the equation? Do the calculation you should be able to do. It's just plugging things in and you calculate it correctly. Question is, how do we arrive at these particular numbers?
Put decimals in here, is that fine?
I don't like to do that.
Okay, just for stick around.
So sometimes rounding, it can be weird, but it's not. When it comes to grading the exam, I'm not gonna grade it wrong or right, because it doesn't matter. I'm more looking at the right answer first. If you get the right answer, you probably did things the right way. If you got the wrong answer, then I need to backtrack to see what you did wrong and how much partial credit is that worth. But yeah, sometimes when you're using decimals, rounding errors can come into play. But typically, if you wrote 102 rather than 101, maybe I got that right, or if it's 100 versus 101, maybe that'd be okay, or maybe just be a small deduction. But I might exam this for 4 things, 4 digits, just sort of count for, are there any rounding errors somewhere or the other? Yes.
How did you get the 3/8?
So if you look at trying to do a Punnett square, lavender, because of what it says, purple is dominant, lavender intermediate, white recessive. The fact that there's 3 phenotypes means that's an incomplete dominance example. So we take our B gene, 2 heterozygotes, and do the Punnett square. We separate the capital B from the lowercase b. And so in this example, this homozygous dominant is the purple. These 2 are lavender because they're the heterozygous case. And this one is the one that would be white. Can't see it. Blonde. And so one half of the B's will be lavender, 1/4 will be purple, 1/4 will be white. And for green, we do the same thing. We have heterozygous parents. And do our Punnett square, we separate the capital E from the lowercase e for each parent. Combine them back together. Because this is a case of complete dominance, the homozygous dominant and the heterozygous are all the same phenotype. We get 3 greens, or green is now 3 out of 4. So 3/4 times 1/2, 3 out of 8. Yes.
Like, will we always be told that there's an intermediate?
You'll either be told that it's an incomplete state, or be given something like this where it tells you what are the phenotypes.
Okay.
So based on the 2 phenotypes, you should assume that means complete dominance, 3 phenotypes means incomplete dominance.
Okay.
It sort of stays spaced, but instead of saying, here's complete dominance, and then listing traits, I just list them and say a little space. But sometimes it will state implicitly complete dominance for gene A, or eye color, and then incomplete dominance for flower color, and then complete dominance for leg color. I don't know what you call this. Any other questions about doing this calculation? And this is the format that you'll see in your exam, but it is going to be different genotypes, different traits, different numbers. Typically leading up to this is a question that makes you use the addition rule or the multiplication rule just by itself. Maybe not, because this already makes you do the multiplication rule to get the individual probabilities. So you probably only have 2 questions here using the or statements of the addition rule, and then doing this repeated trial experiment. If you haven't looked at the lecture reviews from last time, there are 4 problems that you can work from 2 different sets of data. You will see this type of problem on your quiz this weekend. Practice before you attempt the quiz. Final questions about doing the binomial equation probability calculation. Anybody thoughts, questions, concerns? Sometimes it's hard to know if you have questions until you go home. Try it for yourself. But let me know. This next part is an extension of the last part. Pedigrees and Punnett squares and probability calculations go hand in hand. It's the same type of idea, but a different type of purpose. The whole idea of pedigrees is to try to figure out what's going on when you can't do genetic testing. Gregor Mendel was able to manipulate plants, do all the follow experiments we can't do with humans because they're icky, or people don't want to have one child, or no children. Not everybody can have octuplets. But if we did, we have a whole lot of good genetic data to deal with. I mean, we're all monocultures anyways, you didn't really realize. Because of, the death that's occurred over the eons. That's why all humans are the same, basically. There's come from very small subset of populations. Anyways, pedigrees. It's not just a dog food. Or is it cat food also? I forget. But one of the problems with trying to track genetic disorders through families is a small number of individuals in a family. Typically, there's 1 or 2 kids, maybe 3, maybe 4, but generally it's a small number. And with small numbers, the variability is very big between 1 positive and 2 positives. So we have to do some other type of thing. And pedigrees allow us to look back through time to see what kind of disorder might be affecting a particular family or unit. So we're limited because of the genetic data that we can get. We can't force matings to occur to see how things are transmitted and what kind of relationship there is between different alleles. Now these days we don't really need pedigrees as much because of DNA sequencing and a lot of genetic testing that can go on. Now some of this genetic testing simply means karyotype analysis. Again, we can easily spot certain types of genetic defects by karyotypes. Other ones we would need to do either a pedigree analysis or to sequence something to see, do you have the allele associated with the disease or not? Trying to identify people as carriers is a pretty important idea. I don't remember if it's still required in every single state, but for the longest time you had to take a blood test or you get married to somebody to see if you are too related to each other. What is the problem if you are too related to each other? Well, inbreeding is an issue for a reason, because it means you get a overexpression, not expression, best word, but that's the best word off my head right now. You're more likely to see recessive phenotypes when you breed with somebody that's similar genetic makeup as you. So that's one of the things we're really trying to look for. Turns out carriers are the bane of any type of analysis. But there are fun things we can do with genetic screening. I always like this one because who doesn't want to shit in a box and mail it to somebody and not be ironic or funny or mean about it? You can either do this or you can get a camera up your butt and they will go around through your intestines. Do funny things with you when you're knocked out, I guess. Something to look forward to. Both men and women must have colon cancer screening to be healthy. If you're mostly a woman, not mostly a woman, I've heard most. Let's rephrase that. I guess if you're a woman, you probably care more about it than men. But men do suffer from breast cancer. It's just not as common. So this is just another company that does genetic screening for certain types of breast cancers. So there's lots of these things you can do out there. It's always good to know what your genetic makeup is. It tells you what are your likelihoods, basically. What is your penetrance number? What's your probability of getting a certain disease? So that's sort of the fun of genetic screening. But this is about pedigrees, and what can we do when we look at a family tree? This slide is exactly the same slide as before. Just so you know, it is really the exact same slide as before. I don't remember why, but it didn't hear me in, other than to remind you that pedigrees allow us to not have to worry about genetic testing. We simply have to look at a data set. But it does require to have a certain number of generations for it to be useful. If you only have 2 generations, you're not going to have a whole lot of information on what type of disorder is present. So the more generations of data you have, the more confident you have in your analysis. When we look at pedigrees, there are 1 of 5 different things we're looking for. These are the types of disorders that are commonly or easily identified through pedigrees, and generally speaking, these are the types of disorders that we will suffer from as humans. Autosomal dominant, autosomal recessive, X-linked dominant recessive, and Y-linked. Now, why is They're not a dominant and recessive for the Y-linked disorders. You only have one Y, so dominant and recessive really have no meaning. Which means Y-linked is usually a very easy one to spot. It only affects men, and it always affects every man in a family group. These are just some examples of common diseases that fall into these categories. I'm not gonna actually memorize any of these, but they're just so much fun to look at. Although I will mention Fragile X syndrome will come up again in a later lecture. But it's not because it's an X-linked dominant disorder, it's because it's something else that's interesting to talk about. Anybody want to play Hollywood Squares? You know what Hollywood Squares is? It's tic-tac-toe with celebrities or contestants. Pick a celebrity, and the host asks the celebrity a question, and the contestant determines, did the celebrity get it right or wrong? Now, if you Google it, it'll say circle gets the square, or X gets the square. Why do I bring out circles and squares? Because that's the symbols we need to think about with a pedigree. And as we all know, men are squares, and women are circles. A circle is a perfect object. A square is not. Could be the opposite, I don't know why. But here we are, squares and circles. It's up to you to remember that the square is the male one, and the female is the circle one, or vice versa. I should stop talking now before I get myself in trouble. When we look at the square and the circle, there are different ways that that symbol can be oriented, and we have open circle, filled circle, and half-filled circle, or square. You can see what each of these represent. Normal versus disease state or affected, and carrier state. So these are the ones we really care about most because they indicate a certain type of disorder very quickly and very easily. And so this is an example of what we will see on your quiz this weekend, and on exam 2, and on the final exam. You need to tell me, what is going on with this family? Is it dominant? Is it recessive? Is it autosomal? Is it X-linked? Is it Y-linked? And how do you know which one's which? I'm going to try to teach you this as we go forward. There are a few that are very easy to think about. The first is Y-linked. Again, only males get a Y chromosome, so only males will be affected with a Y-linked disorder, and it'll be every male. There's no way to not be affected by a Y-linked disorder unless you are marrying into the family, because then you're not going to get DNA from anybody, but that's a different story. And something you also have to look for is the male a offspring or just a spouse. Does those matter sometimes. When it comes to X-linked disorders, we do see there is a difference in the male versus female affected rate. And autosomal disorders, because they're autosomal, we would typically assume that both sexes should be equally affected. Because it doesn't matter if you're male or female, because the X chromosome and the Y chromosome are not involved at all. But when it comes to X-linked disorders, because of the male genotype XY and the female genotype XX, there is going to be some differences that are inherently entwined in that inheritance pattern of the sex chromosomes. Now, by far, one of the easiest things to do is to ask yourself a first question. Is this dominant or recessive? And the easiest way, or the reason why this is the easiest, is because carriers will always tell you if this is a recessive disorder. You ever see any half-filled in circles? You automatically know this is a recessive disorder. The other thing we need to keep in mind, carriers represent heterozygote situations, which is why they're carriers. The recessive allele is hidden by the dominant allele. It also means that this is going to be a complete dominance trait. Can't happen for incomplete dominance, Because an incomplete dominant is both the recessive and the dominant combined together to create a intermediate state. So you can't have carriers with incomplete dominance. Only with complete dominance will carriers show up. That's a fun fact, but not something that's a quiz or exam question fact. It's just something you should sort of realize. The carriers are heterozygous. So an individual will have the recessive allele, but not be affected by a disease, which is why they're carriers and typically unknown and hard to detect without genetic analysis. So we always know whether it's recessive or dominant based off of the presence or absence of carriers. Once we know that, one question we can ask ourselves, is there a difference? So once we know it's recessive, we have to determine, is this a X-linked disorder or a Y-linked disorder? There's a couple things we can look at and think about as we make this determination. Is it autosomal or is it X-linked? And one thing we will always see with an X-linked disorder is males will always be affected by an X-linked recessive disorder. Because they only get one X chromosome, doesn't matter if they get the right one, recessive, they're going to be affected. Now it's not that all males are affected. It's that males are always affected by X-linked recessive disorders. Which is again a weird thing to think about. Because you have two X's you can get from your mom and one Y from your dad. And in a carrier state, it all depends on which one you get. So it's not all males are always affected, just males are always affected. I know it doesn't sound different. Actually, this is not even affected at all. Who wants to come make a dumbass today? You can't do it, you did it right. This is about carriers. You stupid preaching. I was getting ahead of myself. This one's carriers. Males are always going to be carrier. Oh, that's not right either. What the hell is this? What the fuck is going on here? No, you can't be carriers. Yes, that's right. Males cannot be carriers of the X-linked disorders. Females can be carriers of the X-linked disorders. That makes sense. Females can be carriers of both X-linked disorders and autosomal recessive disorders. This will make more sense as we go forward. I should look at pedigrees. Try to do the analysis. But in a recessive disorder, you can never get a case where certain things are going to happen. I'll leave it like that because I don't want to set myself on landmine again. It'll all make sense in just a little bit. You have to trust me on that. By far, one of the most more difficult things to do, which you probably think ought to be more difficult than what I just talked about. Determining whether something is X-linked dominant or autosomal dominant is going to be the harder or the hardest thing that you'll need to worry about doing. There are some basic rules we can talk about, which are that in an autosomal disorder, males and females should be equally affected by the disorder. It's not that you always get a 50-50 or 100% ratio. It's that the pattern that we see is going to fall into a certain category versus a different category. And the easiest way to learn this is to go through some examples, which is what we're going to deal with. Now, when it is an X-linked dominant, we will see a little bit of a difference than when it is autosomal dominant. And again, the reason for this difference is because of the number of X chromosomes present in a male versus a female. And what does a male pass to its offspring versus what does a female pass to their offspring? We can see certain patterns emerge as we look at the data. Again, looking at the data, looking at pedigrees, allows us to think about these things a little bit more deeply. So as we look at our pedigrees, we need to ask ourselves some questions. The first question is always, are there carriers present? After we ask ourselves that question, we ask, are there male carriers And are they bringing your thing from Amazon? That's a pun. Male versus male. We call a homophone. Things that sound the same but are spelled different. And then are there female carriers? Or are there both? These are things we want to think about. So let's look at this pedigree on board. Do we see carriers or no?
Yes.
There are, again, circles in there that are half filled. But there are symbols in there half filled. So yes, the answer is yes. We have carriers, which means we are looking at a recessive disorder. Now we have to decide, is that recessive disorder autosomal or X-linked? Now in a recessive disorder, females can be carriers of X-linked and they can be carriers of autosomal. These are possible. For a male carrier, the only time that you can be a carrier of an X-linked disorder, or never X-linked, males can only be carriers of autosomal and never X-linked. So now we know there's carriers, we just have to identify what types of carriers are there. And look for female carriers. Because if it's female, we know it can't be X-linked. Or if there are no male carriers, we know it can't be X-linked. And in this, do we see any male carriers? Nobody's bringing packages from Amazon or wherever you order things from. Food Fresh or Hello Fresh. But there are female carriers. So we know it's not X-linked because there's no male carriers. And they've already eliminated the X-linked as a possibility, which means just by default, the fact that there are carriers in here and the only carriers are females points us towards it being, sorry, that way. Points us towards this. Now the other way to look at this is, can we do a Punnett square? Can we think about what is going to happen when we look at our individuals? I really feel like on Friday I'll just be going over this whole thing again, because I screwed it up unintentionally. But if you look at autosomal versus X-linked. Let's look at any group here. We'll look at this right here. We have a female and a male. If it's autosomal, the carrier means that we are recessive, and the male means that we are what? What would be the genotype of this particular individual here? It have to be homozygous dominant. They can't be heterozygous because that would make it a carrier state. It can't be homozygous recessive, because that would be an affected state. Because they were determined it's a recessive disorder, and for recessive disorders, it has to be a homozygous recessive genotype to get the disorder. So if we assume or think that this is an autosomal possibility, this would mean what our genotypes of our two parents would be. In which case, if we do this Punnett square, we should get no people that are affected, but it should get some that are carriers. And does the data show that? If that doesn't show any carriers, it does show some affected. So just by looking at the Punnett square as a possibility, we should eliminate this as a possibility because A, there should be males and females affected, but small number sizes, some variability, you don't know if you're gonna see everything. What we do see is that the male is affected and all the females are not affected. This probably indicates there might be a sex component to this, which is why X-linked is most likely the case. So if we assume it's X-linked, we have female, it's going to be basically big X, small x, just for dominant recessive. The male is unaffected, so it's big X and little y, just for fun. And if we take big X and small x, do our Punnett square and take big X and y, we get big X, small x, x, y, and then we get a little y, x, y. So females can be unaffected, males can be unaffected, female can be a carrier, male would be affected. So in this Punnett square possibility, we see the things that are possible. We have unaffected female individuals, and we have an affected male individual. That's the thing we want to keep in mind. Also, is that women only give X chromosomes to their sons. So as we're looking at an X-linked disorder, what is the mom giving to the son in case it's a carrier? If only the sons are affected, and all sons are affected, you should think that that is a X-linked disorder case. Now, based off the genotype and the Punnett square, there are going to be some that are unaffected. So some that you're always affected when you're a male, because you have the possibility of the good X chromosome from your mom. So we need to sort of try to think backwards. What are our possibilities? Now, a lot of times with the pedigree, there's only one or two that are going to have useful bits of information, so you kind of have to guess sometimes what's going on here. We definitely know it's recessive because of the carriers, and based off of the pattern of carrier mom leading to affected males, should point us in the direction of an X-linked disorder. And generally speaking, the easiest way to do this is female carriers can only, or if there's only female carriers, that indicates it's an X-linked disorder. Because if it were an autosomal recessive, there should be some male carriers in there. And a lot of times, what you should see should be enough information to make you choose one way or the other. I'll try one more, see how we do. Sort of walk you through that one. Okay, first question is, are there carriers?
Yes.
So actually, I know you're gonna hate me about this, I'll just change this real quick. If there's only female carriers, that is X-linked. If you get both, male and female carriers, then it could be autosomal. So in this case, we saw carriers, so we're recessive, and what do we see as far as the pattern goes? Are there, what kind of carriers do we see? And so we see both examples of male and female carriers. This is a weird case where you have two lines between somebody, that means there's a genetic relationship there. This is cousins, which, you know, first cousins don't just have sex with each other and have children. There's two genetically similar. So two carriers, when you have sex with somebody and breed with somebody that's genetically related to you, you're more likely to see autosomal recessive disorder show up in your children. Yes, based on this one, we should say, yes, it is recessive. And the fact that there are male carriers in this pedigree should make us say this is an autosomal disorder and not simply an X-linked disorder. Again, one of the more difficult ones to deal with is the autosomal ideas. Sorry, the dominant disorders. In this case, we have that case here. Again, no carriers means this is a dominant disorder. We need to try to decide, is it an X-linked dominant or a Y-linked dominant? And one of the ways that we can do this is if it is an X-linked dominant disorder, are males or females going to be affected? Because you get X from mom, but not from dad. And so that's the thing we want to keep in mind is the inheritance of the X chromosome. You get some from mom, some from dad. So males will only inherit X chromosome from mom, daughters will inherit X chromosome from dad. So we look at any of these patterns here. A dad is giving an X chromosome to this daughter and a Y chromosome to these sons. If it were an X-linked disorder, this should be affected because it's getting the affected X chromosome from the dad, and the dad only has one X chromosome. It has to give it to the daughter. So it can't be X-linked, otherwise this daughter would be affected. And basically all daughters would be affected if the male is the affected one. So we're trying to sort of follow a pattern. So if we say no carriers, we'll definitely have to start again with this one, right? Finish this up. So no carriers means it's dominant and easy, hopefully, for everybody to think about. For affected males, all daughters should be affected. If it's excellent. And arms that time, so we'll have to pick up for the other part. But if you can eliminate this possibility, then you're only left with one thing, which is autosomal dominant. But we'll continue this.